Good morning, everyone. It's a great pleasure to be here today to present our work on RA CRISPR knockout screening human intestinal epithelial organoids in the Functional Genomics Screening Laboratory. And this is the first screening project we successfully completed in our lab and within a year since we launched. I'd like to start by giving you a short introduction on the lab and the unique collaborative framework we work within. So the FGSL is a cross-functional partnership between the Medical Research Council, AstraZeneca, and the Milner Therapeutics Institute based at the University of Cambridge. And our lab, which is based at the Milner, is set to perform R8 CRISPR knockout screens where individual gene perturbations are tested in separate wells to study gene function and link this to a disease-relevant phenotype. We collaborate with UK-based academic researchers and small and medium-sized enterprises, and we predominantly focus on primary and stem cell-derived in vitro models that fall outside of oncology. Our lab is equipped with an advanced automation platform, which gives us the ability to perform large-scale screens up to genome-wide capacity. And in this video, I hope you get a glimpse of how the different components work together. We use liquid handlers for media replenishment. This is the Bravo. And we also use the acoustic dispenser, the Echo, in order to transfer guide RNA library or Cas9 directly to the assay plate with the use of sound waves. The CertusFlex dispenser could be used to perform compound treatment and antibody staining. And the robotic arm links all these instruments together and automatically transfers one plate from one station to the next one. So by automating this workflow, we don't only reduce the risk of human error, but we also streamline a highly laborious process. The platform, when we run the screen, is controlled through software and requires minimal user input. And while not shown in the video, this platform is compatible with imaging-based, plate reader-based, and also flow cytometry-based readouts. So the work I'll present you today is a result of collaborative effort from various teams. The project was led by Ulrike Kunzel and Chun-Hao Wong, and Taxiaris and myself performed experimental work with the help of Ben setting up the automation. Anka led the bioinformatics analysis, but we also benefited from the expertise of our Milner colleagues, Tom and Erica, who provided expertise on gene editing and the model itself. Also from our AstraZeneca collaborators, who gave us extensive expertise along the screen, and also the academic lab led by Matthias Ilbauer, which is based at the Stem Cell Institute, who provided the model, disease expertise, but also the gene list for the screen. So dysfunction of the intestinal epithelium is a key driver of inflammatory bowel disease, including Crohn's disease and ulcerative colitis. And despite this, available treatments do not directly target the epithelium, are effective in only 50% of the patients, and largely focus on symptom management rather than curing the disease. Emerging evidence suggests that epigenetic mechanisms may play a role in IBD pathogenesis. However, the mechanisms that drive this remain unclear. Our collaborators recently showed that loss of DNA methylation at MHC class I loci is a hallmark of the intestinal epithelium. So in order to study this a little bit further, we use human intestinal epithelial organoids derived from pediatric patients, a model that has already been established by our collaborators group. This retains donor tissue-specific information and also recapitulate inflammatory responses observed in patients, such as MHC class I activation, when they're exposed to IBD-relevant inflammatory cytokines in vitro, like the interferon gamma. So using this physiologically relevant model, in this project, we set to perform an R8 CRISPR knockout screen to identify novel regulators of MHC class I in the intestinal epithelium, with the ultimate goal of uncovering new therapeutic targets for IBD. Our screening approach utilizes a pool of 3 guide RNAs per gene, complex with Cas9 protein in order to form a ribonucleoprotein complex, which is delivered to the cells via electroporation in 96-well plate format. Among the genes we targeted were genes involved in epigenetics, in MHC class I pathway, and IBD susceptibility. All these informed from previous data obtained by our collaborators. So after gene editing, we plate the cells into the grid 3D imaging plates, which I'm going to cover very shortly. We let them to grow. And then 5 days later, we perform interferon gamma treatment in order to stimulate MHC class I activation. 2 days later, we fix and stain the organoids using a nuclear stain, a marker for the epithelium, and also a marker indicative of MHC class I. Then the plates are processed for high-content imaging in our spinning disk confocal microscope, and the results are analyzed using the commercially available software, Signals Image Artist. In our gene editing experiments, we include neutral controls. In this case, we use the AVH1 safe harbor locus. So this introduces a cut in the double-strand break cut, but doesn't produce a phenotypic change. And we have this without interferon gamma and with interferon gamma stimulation in order to establish a baseline response and a response to interferon gamma treatment. Also, for positive control, we included the P2M, which is a gene that encodes an MHC class 1 subunit, and this was used to validate the assay performance. And what we call a hit is basically any gene knockout that produces a significant phenotypic change compared to our controls. So as I mentioned, a key aspect of our experimental workflow is the use of microcavity plates in order to culture and image the organoids. These are plates where each well contains an array of microwells of a defined diameter that allow the organoids to grow in a confined space and in a controlled manner. Each microcavity has a U shape, as you can see here, and is made of PEG hydrogel, which is optically clear, making these plates imaging friendly. Also, due to the fact that the organoids grow quite separately, these plates help a lot in the downstream analysis and in the segmentation of the organoids. Another advantage of these plates is the presence of the pipetting port that you can see here on the side. This enables safe media exchange without disturbing the layer where the organoids grow, and is highly compatible with liquid handling systems, so it was very nicely integrated in our automation platform and workflow. As part of the assay development, we wanted to establish the interferon gamma concentration that could reliably induce MHC class 1 activation. And in order to do so, we treated the organoids with two different concentrations of interferon gamma, 0.2 and 2 nanogram per ml. Then we fixed the organoids and we stained them with an antibody against the HLA-BC as a readout for MHC class 1, because these are the classic human MHC class 1 molecules. Our experimental results showed that there was a dose-dependent increase in HLA-BC levels in both in two donors that we tested, and since donor 1 showed a greater response, it was the donor that we selected for the screen. Another key part before moving to the screen was optimizing the dynamic screening window, and this is basically to define the conditions that a gene count could reliably and significantly reduce the HLA-BC levels after interferon gamma stimulation. We first tried to adjust the Cas9 to guide RNA ratio. I'm not showing this data, but what we found it worked better for us was increasing the amount of Cas9. So alongside our controls, the AVH1 and the P2M, we also included interferon gamma receptor 1 as an additional editing control. After interferon gamma stimulation, we see a nice, strong increase in HLA-BC intensity, as you can see with the magenta color compared to the gray color, which is the non-stimulated. In contrast, knocking out P2M led to a reduction in HLA-BC levels, even below the baseline levels. More importantly, increasing the amount of Cas9 from 5 to 7.5 improved the effect of interferon gamma receptor 1 knockout and led to a stronger decrease in HLA-BC levels, as you can see in the plot here, but also in the example IF images on the side. So based on this, we decided on the RNP conditions, and also we selected the positive controls for the screen. So having defined the guide RNA delivery and also implicated the necessary controls, we also incorporated automation in our workflow. I won't have the time to go through it, but if anyone is interested, I'm happy to talk to you about it during the poster break. Our poster is 58. So to ensure robust data collection, we carefully designed the plate layout, and we strategically distributed our target genes and control genes across the plate map. We screened in total 140 genes in 2 electroporation plates with 3 replicates per plate, and also we did that in 2 different donors. With the different colors, you can see our different controls. So we first asked how robust was the assay and whether we could reliably call hits. So looking at the quality control metrics, as you can see here, our technical replicates from this example, they're highly consistent. Also, we have a really small variability within our control groups, and also a very nice and clear separation between our neutral and positive controls. This defines the assay window. So this clear separation relative to variability is captured by the robustness factor, and this is a standard assay quality metric used in high throughput screens. Across the screen, we got an average robustness factor of over 0.3, which is the cutoff in high throughput screens in these models, which gave us confidence in our screening results and led us to the heat calling phase. So any genes whose knockouts fall within this assay window could be considered potential hits. So with confidence in the assay quality, we can now look at the screening output and our results. The heat map on the top summarizes the hits ranked by observed effect size for MHC class 1 measurement using a cutoff of 0.3 in either direction. With blue color, we see knockouts that led to a reduction in MHC class 1 levels, whereas with the orange, we see hits that led to an increase in MHC class 1. With white, we have hits that had no effect in one of the two donors. So it's quite clear that we have two different clusters, one where the hits are served between the donors and one that the hits appear to be more donor-specific or had a moderate effect in one of the two donors. So knowing that, we wanted to see how reproducible are hits across the two donors and whether we can quantify that. So we plotted the effect size of hits in MHC class 1 in donor 1 against donor 2. In this scatter plot, you can see our normalized data where the neutral control sits at 0 and the positive control sits at -1. The dashed lines correspond to the 0.3 or 30% effect size, which was set as a cutoff. So the positive correlation, as I hope you can appreciate that most of the points fall close to this diagonal line, suggests that our hits are largely consistent across the two donors. And in total, we found 12 hits that serve between them. So during our image analysis pipeline, we also managed to extract some additional information, and this was the health status of the organoids. These health scores were obtained using a machine learning module trained on images acquired from healthy controls and from genocounts that gave us a lethal phenotype. So the heat map here summarizes the health score of the different genocounts that correspond to the hits on the top. And this is particularly useful when you are considering on taking screens further for target validation. So I hope my presentation gave you a taste for how from proof of concept we got to a screen in around a year. I show you some data regarding the optimization of the guide RNA delivery. We also optimized the endpoint assay, and we performed the screen in 2 different donors with good, robust assay performance and also results that gave us confidence in our heat calling approach. We developed a semi-automated workflow for RNA perturbation in the organoids, and also we developed an image analysis pipeline and also data visualization tools that will be applicable in other organoid models. The next steps would be to validate the top hits in additional donors and using orthogonal assays, which is something that will be followed up by our collaborators outside of our lab. That brings me to the end of my talk. I'd like to thank you for your attention, but first I'd like also to thank the lab members of our group who helped us shaping this project, our collaborators, the MEINER, which is the institute that hosts us, and the lab of Matthias Silbauer, who gave us extensive expertise on the model itself. If you're interested in hearing more about our work or collaborating with us, please visit us at our booth or step at our posters 10, 58, and 50. And last but not least, as John mentioned, we are launching a seminar training development program. This is designed by ECRs for ECRs and it's part of the initiative. So if you want to be part of it, I think it's a great opportunity for ECRs to present their work, but also support each other. Please join our community, scan the QR code, or come and talk to us at the ECR table. Thank you very much for your attention.